Good morning, students, and welcome to today's class on blood and its component and functions of blood. So, blood, it is a red colored fluid connective tissue which is fluid in state, that means it is liquid in state when it remains inside the blood vessel, but when it comes out of the blood vessel it clots or it converts into some jelly like substance which is called as clot. So, volume of blood in adult human being is approximately 5 to 6 liters, and it is slightly alkaline in nature, having a pH in normal condition at 7.4. Blood has majorly two components. One part is the liquid part which is considered or which is called plasma. The other part include formed elements, that means blood is consisting of two parts, plasma and formed elements. Plasma constitutes 55% of the blood and the formed elements, they constitute 45% of the total blood volume. The formed elements include various blood cells like RBC, WBC and platelets. It constitutes 45% of total blood. So, plasma, first part of the blood, first constituent of the blood, that is plasma. So, plasma, it is straw colored fluid which contain approximately 90% of water. As the plasma is liquid in form, so it contains 90% of water. Also, it has approximately 7% plasma proteins. So, the proteins which are present in plasma, they are majorly of the class albumins, globulins and fibrinogen. So, albumin, globulin and fibrinogen type of plasma proteins are present in plasma, which constitute 7% of total plasma. Then plasma also contain 1 to 2% organic substances and 1 to 2% inorganic substances. Also, it contains dissolved respiratory gases like oxygen and carbon dioxide. Plasma without fibrinogen, as we have already stated that plasma proteins are of three types, albumin, globulin and fibrinogen. So, if the plasma is without fibrinogen, then that type of plasma is called serum. So, only difference between plasma and serum is the fibrinogen. If it contains fibrinogen, then it is plasma, and if there is no fibrinogen, then the plasma is itself is called as serum. The organic substances which are present in the plasma, it includes carbohydrate in the form of glucose, then natural fats and phospholipids and cholesterol, then nitrogenous substances like ammonia, amino acid, creatine, creatinine, xanthine, hypoxanthine, urea and uric acid. Secretions are also present in plasma, because as we know that the blood is a circulatory fluid which circulates throughout the body. So, it also carry the secretions of various glands like hormones and antibiotics which are secreted from various parts of the body, and that they are carried to their target objects. Also, the enzymes are present in plasma, that are the organic substances. These enzymes are like amylase, alkaline phosphatase, acid phosphatase, lipase, esterases, proteases and transaminases. The inorganic substances are in the form of ions and salts of various compounds, like the ions and salts of sodium, then calcium, potassium, magnesium, chloride, iodine, iron, phosphate, copper, etc. So, all these type of substances, they are present in plasma. Now, the second half of the second component of the blood, that is blood cells, or you can say formed elements. Now, formed elements which constitute 45% of total blood, it includes mostly the blood cells, or you can say blood corpuscles. It include red blood cells which may be called as erythrocyte, or you can say red blood corpuscles, then white blood corpuscles or white blood cells, or may be called as leukocytes, and third one, that is platelets, or may be referred as thrombocytes. Now one by one we are going to study in detail all the three type of blood cells. Firstly, red blood cell. Now RBCs, in short they are referred as RBCs. So RBCs are circular, small and biconcave in shape. Their shape is circular, they are smaller in size and they have biconcave surface. It is just like a disc which is a concave on both sides. So biconcave surface. Non-nucleated in all mammals. RBCs are generally non-nucleated in all mammals, but only the camel has a nucleated RBC. Remember, the camel has nucleated RBC. Rest of all mammals, they have non-nucleated RBCs. The process of development and maturation of RBCs inside the hemopoietic organs, that process is called erythropoiesis. The process of development and maturation of RBCs is called as erythropoiesis. The organs, or you can say the hemopoietic organs which produce RBCs, they are liver, spleen, lymphoid organ, and these organs produce RBC during prenatal life, while in adults the RBCs are produced from red bone marrow cells. Average lifespan of RBC is approximately 120 days. So 120 days is the approximate lifespan. They remain active during this 120 days. Red color of the blood is due to the presence of hemoglobin in the RBC. So RBC contain this respiratory pigment which is hemoglobin. RBC count in adult human male is approximately 5 millions per cubic mm of the blood, while in females it is about 4.5 millions per cubic mm of the blood. Increase in RBC count is called polycythemia, while the marked reduction, that means the demarked reduction in RBC count, may be referred as anemia. So anemic condition, if the RBC count decreases, it is called as anemia. Now second type of blood cells, that means white blood cells or white blood corpuscles, may be called as leukocytes. So leukocytes, they are colorless nucleated cells. They are rounded or amoeboid in shape, larger than RBCs. Their size is larger than RBCs. They measure about 8 to 15 micrometers in diameter. However, the RBCs, they measure approximately 7.2 or 7.4 micrometer in diameter. So these are larger than RBCs. They are capable of amoeboid movement against the bloodstream. That means they can move in the opposite direction of the bloodstream. Bloodstream is flowing in one direction, so these WBC are capable to move in opposite direction. They can pass through capillary wall by the process called diapedesis. In the diagram here you can see, the process by which WBC are coming out of the capillary wall. So they can pass through this capillary wall, and this movement is called diapedesis. This occurs when there is need of phagocytosis outside the blood in the tissue. So these WBC, they can move out of the blood capillaries and they can kill the pathogens outside the blood, and this process of passing through the wall of capillary is called as diapedesis. Total number of WBC in the blood is approximately 4,000 to 11,000 per cubic mm of blood. They are produced in bone marrow, spleen, lymph node, tonsils and Peyer's patches. Leukocytes are classified mostly into two groups on the basis of cytoplasmic granules, whether the granules are present in the cytoplasm or they are absent in the cytoplasm. On that basis, the leukocytes are classified into two types. One is called granulocytes and second is called agranulocytes. Firstly, we will see the granulocytes. The granulocytes constitute 65% of total WBC of the blood. These granulocytes are with cytoplasmic granules, or you can say the WBC with cytoplasmic granules are called granulocytes. In the figure, various granulocytes are shown having the cytoplasmic granules. Some granules are larger and coarse, some are fine and small. They are also called polymorphonuclear leukocytes because their nucleus is polymorphic. That means the nucleus is in variable shape. Hence, they are referred as polymorphonuclear leukocytes. They are classified again into three types. One is neutrophils, second one that is eosinophils, and third one that is basophils. Now let us see one by one these three types of granulocyte. Now neutrophils. Neutrophils, as you can see in the diagram, they have a multilobed nucleus. They are approximately 60% of total WBC. Their nucleus is multilobed and they are phagocytic in function, and they usually form the pus when there is acute infection. Then second type of granulocyte, they are called as eosinophils, also they are referred as acidophils. Now remember that the cytoplasmic granules in neutrophils, they accept both type of stain. When these type of WBC are stained by a particular biological stain, they accept both type of stains, that means acidic stain and basic stain. Hence they are called neutrophils. Now second type, that is eosinophils or acidophils. This type of WBC, they are only 1 to 3% of total WBC, but their nucleus is bilobed. So in the figure you can see there are only two lobes of nucleus. Here this is one lobe and this one is second lobe. So there are only two lobes. So nucleus is bilobed, but the cytoplasmic granules of this type of WBC, they only take or they only accept or they are only stained with acidic stains. That means acidic dyes can only color this type of WBC. They can only give color to this type of WBC. Hence, as they are accepting only acidic stains, so these are called as acidophils or eosinophils. Their function is anti-allergic and they produce antihistamines. Also, they kill the large parasites. So, anti-allergic and antihistamine properties are there in acidophils. Then third type of granulocyte, that means basophils. Now basophils, the name itself indicates basophils, that means the cytoplasmic granules of this type of WBC, they take basic stain or they are stained with basic dyes. It is about only 0 to 1%, that means they are rarely seen in the blood. They may be present 1 in per cubic mm of the blood. So approximately 0 to 1% of total WBC they are found. Nucleus is roughly S shaped. They produce histamine. They also produce heparin and serotonin. Then second part or second type of WBCs, that means granulocyte. So agranulocyte, the name itself indicate means without, granulocyte that means granules in the cytoplasm. So these are the WBC without granules in their cytoplasm. They are approximately 35% of total WBC. They are non-granular WBCs. They are classified again into two types. One is called monocyte and second one is called lymphocytes. Now one by one. Firstly, monocytes. So monocytes, these are about 3 to 5% of total WBC and they are larger in size. They are larger in size. They have kidney shaped nucleus. The figure shows kidney shaped nucleus in WBC. Then they are highly motile and phagocytic and they remove damaged tissue, hence they act as a scavenger cell. They are also called big eaters because they are big in size and they have phagocytic property, so hence they are referred as big eaters, and they remove the damaged tissue from the cells or blood, so hence they act as a scavenger cell. Then lymphocytes. Now lymphocytes are 25 to 30% of total WBC and they are oval or rounded. They have oval or rounded nucleus with very less cytoplasm. They are of two types, one is B lymphocyte and second one is T lymphocyte. But prior to this, we can say lymphocytes are of two types, smaller and larger lymphocyte. The larger lymphocyte, whenever necessary, they break into smaller lymphocytes. So according to their maturation site and its occurrence, they are classified into two types, B lymphocyte and T lymphocyte. B lymphocyte mediate humoral immunity, that means it produces antibodies and antitoxins. The T lymphocyte mediate cellular immunity, that means it kills the antigen by phagocytosis. Now third one, that is platelets. Platelets or thrombocytes, these are the fragments of multinucleated cells. They are irregular in shape and they are important for clotting. They are very useful in clotting. The normal platelet count in a normal human is approximately 1.5 lakh to 3.5 lakh per microliter of the blood, and it initiates clotting cascade by clinging to the broken vessel wall. Whenever any injury takes place at the broken side, they get attached or they get blocked, and hence they prevent the further flow of blood through that injury. This is the process of development of platelet, how the platelet is formed. Platelets, they are generally formed from megakaryocyte, but the megakaryocyte in turn is produced from promegakaryocyte, which is a advanced form of megakaryoblast. So the platelet formation is initiated from hemocytoblast, which is a stem cell. This hemocytoblast then converts into megakaryoblast. Megakaryoblast then modifies into promegakaryocyte. Promegakaryocyte then gets you can say developed into megakaryocyte, and this megakaryocyte then breaks into thousands of pieces called platelet. One megakaryocyte approximately can produce 2000 platelets. Now lastly, the function of blood. Nutritive function. Generally, blood carries nutritive substances like glucose, amino acid, lipids and vitamin. They are derived from digested food and absorbed from gastrointestinal tract, and are carried by the blood to different part of the body for growth and further production of the energy. Second function is respiratory function. The transport of respiratory gases takes place through the blood. The O2 is carried from alveoli of the lungs to different tissues, and CO2 is carried back to the alveoli from various parts of the body. And third function is excretory function. So various waste products which are produced during metabolic activities in the cell and tissue, they are removed by the blood and they are carried to the excretory organs like kidney, skin and liver, etc. Then transport of hormones and enzymes. Hormones and some of the enzymes, they are carried by the blood to different target organs of the body for the source of secretion. Blood also regulates water balance of the body. The plasma proteins in the blood and hemoglobin act as a buffer and helps in regulation of acid-base balance. Blood also uniformly distributes the heat throughout the body which is produced during metabolic activities. Hence, it helps in maintaining the thermoregulatory mechanism and it keeps the body temperature uniform at every part. So, balance between heat loss and heat gain is maintained by the blood. Then storage function. So, blood serve as a storage of source of water, proteins, glucose, sodium and potassium ions. These substances are taken from blood during conditions like starvation, fluid loss, electrolyte loss, etc. Also, blood plays defensive function, important role in defense of body. The WBCs, which are also called as the soldiers of our body, they are responsible for defensive mechanism of the body, especially neutrophils and monocytes. They are phagocytic in function, so they engulf the bacteria, and the lymphocytes which are involved in immunity. Acidophils are responsible for detoxification, disintegration and removal of foreign proteins. So this is all about the part of blood.